Okay, well, good afternoon. This is just a brief webinar to try and showcase how doing a degree with us at UEL, at the Department of Biosciences of the School of Health, Sports and Biosciences, and actually, irrespective of what degree you choose to do, will help you understand better, gain knowledge, and potentially gain employment after completion of your degree in an area where people investigate the causes of cancer and how to potentially treat it better. So that's what the following half an hour will be dedicated to. My name is Sofia Semerjeva, and I'm one of the subject area leads at the department. I'm responsible for the biological and biomedical sciences courses, which include biomedical science, medical physiology, and biochemistry and biotechnology. With me are the other cluster lead, Dr. Mukhlisur Rahman, who is going to briefly introduce himself towards the end when he's going to take over and speak about what his sort of area of drug and chemical sciences does and how it's kind of also involved with cancer and understanding and particularly treating cancer. So how is this session going to go, or what we're going to do? First, we're going to briefly talk about what cancer is, and I'll try to make this as easy to digest and understand as possible. And then in the latter part of the conversation, I will try and showcase and tell you how doing every one of our degrees, what exactly would you do and how you are going to learn more and potentially be able to work in terms of understanding and treating cancer after completion of these degrees. And at the end, if there are any questions, we'll be able to answer those. So to begin with, I was hoping to have audience and ask what cancer is, but I'll have to kind of answer my own question. And cancer is a very complex disease that actually boils down to a very sort of basic property of actually being able to divide when they shouldn't, and that's why we get cancerous cells and we get tumors, and potentially they spread and become metastasis. So from that point of view, although it's a very complex disease with very many facets, cancer boils down to abnormalities in the regulation and execution of the division of the cells, which is a process whereby a mother cell divides to produce two daughter cells that are identical to the mother cell initially. Now, the two main properties of cancer, or the cells that become cancerous, are that they can divide uncontrollably, i.e., they don't sort of heed the messages that are telling them to stop dividing, or after they divide, the cells produced by them, rather than starting to specialize, continue to divide. And this ability to divide uncontrollably is basically due to the fact that cells that become cancerous lose the balance between how much division of cells should occur to be able to sustain the body, where need be grow, where need be just maintain and sort of repair if required, and how much the cells should actually specialize to be able to sort of do some specialized functions, such as you know, the nerve cells are specialized on communication with the environment, whereas the muscle cells, for example, are specialized on ensuring that our bodies move. So that's one main property or feature of cancer cells, or cancers in principle. The other main feature is their ability to spread from the place where they have initially basically been located and produce metastases that can sort of disperse throughout the human body. And this is what is shown in this image here, whereby you see a metastatic cancerous cell mass basically separating from the place where it has been, or it has originated, and being ready to move to a different place in the body to produce potentially secondary sort of cancerous growths. Now, let's briefly talk about the types of cancers, because I'm sure all of you would have heard about, different cancer names, and so it might get a little bit confusing. But there are three main types of cancers. By far, the largest group of cancers are called carcinomas, with around 90% of all cancers, and they arise from epithelial cells, i.e. cells that have originated from the so-called external or internal cover tissues, epithelia, for the simple reason that it is intrinsic to epithelial cells to maintain ability to divide, because as being exposed, for example, to the external environment, as we're going to see in the next slide with our skin, cells are constantly being shed and lost, and they have to be replaced. So a certain level of ability to divide is being maintained naturally, and is required for healthy life in the human body. The other class of cancers are called sarcomas, which arise from uncontrolled division in the supporting or connective tissues, such as bone, cartilage, and fat. And then finally, we have the cancers of the liquid sort of tissues in our body. Lymphomas are the cancers of the lymphatic system, and leukemias are cancers of the blood, which are uncontrollably dividing, which is what proliferating means, cells that are free in the fluid systems and not and do not occur as solid masses, as was shown in the previous picture. So from the very first year at UEL, the students who study at biosciences are exposed to microscopy and observe different types of cells, both healthy and cancerous cells, and are able for themselves to actually appreciate how at cellular level, using light microscopy, the normal and cancerous cells differ quite appreciably. As you can see, the cancerous cells have very large and often oddly shaped nuclei, shown here in dark blue, compared to the normal cells. Normally, when you look at two types of preparations, one of normal cells and one of cancerous cells, you can see that in the cancerous cells preparation, you see very many dividing cells, because this is a characteristic feature of cancerous cells. They really divide uncontrollably. And there's also disorganized arrangement, whereas you see normal cells, they kind of neatly stay attached to each other, because they would have been produced by division of a cell, and the daughter cells would stay close to each other after the division has been complete. You can see also and appreciate that there would be a variation in size, say much larger here compared to a normal cell, but also the shape can be quite odd. And then you can see that there could be an altogether complete loss of normal features characteristic for healthy normal cells when you look at cancerous cells. So as I said, students doing a degree or any course at our department do have the opportunity from their first semester when they study cell biology to observe such cells and be able to appreciate these distinctive different features for themselves. So we return back to the question of what is cancer, and we take an example of skin cancer. And as you have hopefully remember, the any cancer of the skin would fall under the category of carcinomas, and I'm sure most of you have heard about melanomas. However, these are not the most sort of common types of skin cancer. The most common type of skin cancer is the so-called basal cell carcinoma, and then we have another common type of skin cancer, squamous cell carcinoma, and you would now be able to appreciate what that means. So if you look at the structure of the skin, in a healthy individual, you can see that there's this basal layer of dividing cells that produces all the cells that move externally from that layer, which, as we say in a kind of biological terminology language, migrate or move from that basal layer where they have been produced by division of these basal layer cells towards the exterior of the body. And as they move, they actually acquire specialist features or differentiate. They become specialized. So when they reach the surface of the skin, can you see how very different they are from the cells that are lying just above the basal layer of dividing cells, which are pretty much cuboid rather, because they're three-dimensional. These cells are very much flattened, and they're called squamous cells, and they cover the very surface of our skin. And because we're constantly in contact with the external environment, these cells are subject to, they're the first one to come in contact with any sort of influences from the environment, including actually, humidity or dry air, or UV radiation from the sun, or any chemicals that we come in contact with, and they constantly die. Okay, they're shedded constantly, which is why the basal cells maintain their ability to divide and produce new differentiating cells that become squamous cells. So in a healthy individual, a basal cell will divide and produce two cells shown here. So one of these two cells will maintain the ability to divide, yeah, but the other one would move externally and start to differentiate and eventually will become a squamous cell. That's what's happening for every one of these basal layer cells, okay? What happens when we have a tumor developing, which in this case would be a basal cell carcinoma, because it will be a tumor of the basal cells, is that, yes, one of these basal cells divides and produces one dividing cell, but one that would differentiate. But then the second dividing cell again divides, and rather than produce one dividing cell and one differentiating cell, it produces two dividing cells, okay? And then both of these dividing cells further divide and may produce also two dividing cells themselves. So if you count the number of cells in a normal sort of healthy individual resulting from this one cell after 1, 2, 3, 4 divisions, you would see that the number is much smaller than the number of cells that results here because of abnormalities of the features of the resulting cells, whereby they've actually not listened to their program to start differentiating and move externally and become squamous cells, but they kept dividing, which is why there's an accumulation of additional mass of cells resulting, and you can see this as a formation of a tumor, okay? Now, that's not terribly problematic just yet, because some tumors are benign and they stay where they're meant to be, and they don't cause too much of a problem potentially if they're just small excessive growth. The biggest problem becomes when a tumor becomes malignant and starts to invading neighboring tissues. For example, they could potentially break this basal lamina here, which separates two types of tissues, and then they can invade the underlying tissues. Or worse yet, they may detach, as was shown in one of the previous pictures, become metastasis and move to a different sort of place in the body and instigate development of cancerous growth and tumor elsewhere. So how do we study and understand why is it that some cells do not listen and stop dividing and start differentiating, but they continue to divide? So one way of doing that is by using the so-called cell culture. which literally means that we either take cells from a piece of human tissue taken from an organ. Of course, we'll aim to do this uninvasively, but currently mostly we have banks of cells commercially available which have been immortalized and can grow in these sort of glass or rather plastic dishes. And they are provided, which is not shown in the images, unfortunately, with media, liquid, containing nutrients and growth factors. So let's look at what would happen in a healthy mammalian cell, a cell taken from a mammal, and we are mammals, as you know. So if we put a few mammalian cells in a dish and provide media with nutrients and growth factors, the first thing that these cells would do is attach to the surface of the dish, which is very important for them to start dividing. And this is what is called anchorage dependence, or the division of these cells depends on them being attached to the surface of the dish. Okay. Now, if they do successfully attach to the surface of the dish, they will start dividing, which is what you'll see in the second picture. Eventually, they will cover the entire surface. And when they form a complete coverage, or single layer, as we call that, they will actually stop dividing, which is known as density-dependent inhibition, or in other words, they will be very sensitive that they have immediate neighbors, and they cannot divide any further, producing new cells, so they'll stop dividing, okay? This can further be illustrated by looking at this following image, whereby if you actually take a spatula, a plastic spatula, and gently scrape away some of the cells in this single layer and create an empty space, the neighboring cells will again start to divide and potentially produce new cells, which would eventually cover that space that has been freed and has become free of cells. But once they cover that space again, they'll stop dividing once again. So normal healthy cells require attachment to the surface for them to start dividing. And then they are sensitive to having neighbors and surrounding cells, so they stop dividing when they're basically surrounded by neighbors on all sides and when the entire surface has been covered. Well, investigating cancer cells in these systems, scientists have discovered that the cancer cells exhibit none of these very important characteristics. First of all, they don't need necessarily to attach to the surface to start dividing. And most importantly, even when they cover the entire surface, they continue to divide and produce layers on top of that initial layer. So basically, they're not sensitive to the fact that they have neighbors, and they should stop dividing once they sense their neighbors on all sides. Now, this image was also chosen because I wanted to make the point that we have a thriving cell culture facility whereby very many of my colleagues investigate different aspects of cell biology, which you may learn more about if you come attend one of our open days, for example, and we'll be able to talk to you about that. But this is something we definitely do here at UEL, and our students benefit from this facility. So why do these cells, it's best to illustrate this with the second trait of cells, the fact that they are sensitive to the fact that they have neighbors. Why do cells actually, how do they sense that? Well, they sense that through the fact that cells communicate with each other, and that's called cell signaling. And you'll hear about that if you come to study with us in your first year, and I will be the person delivering that lecture for you. But I did not want to run ahead of the game and tell you all about it, not least because it's a little bit more complicated than what is shown in this image. I chose to use this image because it basically illustrates the fact that cells constantly exchange information with each other via different substances, molecules that they release, and then these substances either act on their immediate neighbors or can travel some distance and act on more distant neighbors. And some cells can actually even produce substances then when released, act on these same cells. So they kind of tell themselves, give themselves messages, a little bit like you waking up in the morning and looking at yourself in the mirror and telling yourself, you must do that task that you've been putting off for a while today. So it would appear that the mirror is talking to you and telling you to do it, but the actual fact is you are producing the image in that mirror telling you to complete that task. So a little bit like that, we have this sort of also a situation whereby some cells tell themselves by producing messages that then are being delivered to themselves. So there's a whole big area of science that investigates how cells communicate with each other. But it is very important for the sake of understanding that, first of all, for the cells to start dividing, they need to hear messages from their immediate environment, which is the media that, like I said, was not shown in this image, which contains nutrients and growth factors. It is particularly the growth factors that signal to the cells in this situation, telling them, oh, there are nutrients around you, and I'm telling you that you should start dividing because the environment is auspicious, you'll get the food, you'll be able to grow after you've divided, and everything will be great, okay? And then when the cells start to accumulate and produce this single layer, they talk to each other and they say to each other, well, we'll have to stop dividing because A, there's very many of us, and then the nutrients have actually been exhausted a little bit, so we'd better stop dividing, else we're going to start to die and basically suffer because of that. So cell signaling is very, very important for the division of the cells. And as we can see, we'll see later on, it'll be very important for the developments of cancer. But what I'm going to tell you now is something quite easy to understand because there are only 3 basic outcomes resulting from cells or any other molecules in the environment talking to the cells. Okay, so cell signaling has only 3 basic outcomes. There are some variations, but the essence boils down to one outcome is that a cell, based on the communication it receives, will decide to divide and will produce new cells, or it will decide to specialize, or i.e. differentiate and become from undifferentiated cell, say a neuron, or it will actually decide to commit a suicide, or the so-called apoptosis, or programmed cell death. Now, apoptosis is a programmed cell suicide that every cell in our body has a code of how that would occur. And that's just a basic image showing you that from this healthy normal cell, when a cell decides that it has to commit suicide or apoptosis, the cytoplasm starts, or the internal content of the cell starts to separate and produce these sort of pieces called blebs, and the process is called blebbing, and eventually the whole cell disintegrates, and the cleaners of our immune system, the phagocytic cells, engulf and eat all these pieces to keep our environment clean and safe. So another illustration of how we study division and abnormal division and regulation of division comes from model organisms such as the free nematode worm Cenorhabditis elegans, or C. elegans. This is a really fantastic model because the worms have defined and known number of cells when they're adults, which is 9,559 in hermaphrodite worms and 1,033 in male worms. Every gene in these worms has been studied because this was the first multicellular organism, an organism made up of many cells, where the full genome has been sequenced, so we know about every gene in this worm. and it's very widely used to study cell division. There's a little kind of video here that shows you how other labs, not us, have managed to attach fluorescing tags so that when every gene starts to be active, you can see a green bubble lighting up, and that's really, really cool. We use different other models. For example, we use the fruit fly, Drosophila melanogaster, for different other studies, but particularly in terms of cancer, C. elegans has been invaluable. It has been invaluable because it showed what is illustrated in this image. Remember, there's a defined number of cells in the adult worm, which is known, and the fate of each of these cells, i.e. what does each of these cells become, and when they specialize, is also well known. So this is a cross-section of a wild type worm, normal healthy worm. And this is a cross-section of a mutant worm whereby one of the genes has been changed such that a receptor, which is a tool of communication between the cells, has been modified such that the cells are not hearing the message to start committing apoptosis or a programmed cell death. When they don't commit the programmed cell death, what we end up with is these additional structures, which you can see here with the arrows, that are not present in the healthy normal worm. And that tells us that apoptosis is extremely important for maintaining the appropriate structure and supporting the appropriate function in C. elegans, but also in any other multicellular organism, because when apoptosis does not occur, as it should, we get these additional structures. And I'm hoping it kind of reminds you of the fact that that looks like something akin to a tumorous growth. Okay, people received Nobel Prize for the work that they did on these particular mutants for the Nobel Prize in 2002 for physiology and medicine. And that work has been seminal in understanding the regulation of division and leading to cancer understanding. So back to cancer, there are naturally present in every one of our cells genes that are tasked with either preventing cell division when it shouldn't occur, called tumor suppressor genes, or genes that actually are not helping anybody but promote division regardless of the fact that it shouldn't occur, and they're called proto-oncogenes. Okay. And in a normally functioning healthy body, they both function as they're supposed to, and we don't get any sort of tumors growing. When we have abnormal division or malignant cell division, the tumor suppressor genes lose their function, so they no longer prevent unregulated cell division, whereas the proto-oncogenes gain function, they support, they drive division even harder, and that's how we can get more cell division and cancer occurring. If we go back to signaling, in order for division to occur, remember, cells that are supposed to divide get information or communication from a growth factor that could be released by a neighboring cell that attaches to a receptor, triggers a number of events inside the cell that tells that cell it should start dividing, for example, when you have to repair a wound, okay? And how would cancer cells actually divide without all these instructions? So first of all, the cells might produce their own growth factor telling them to divide. So they tell themselves to divide when they shouldn't, okay? That's one situation. Another situation is that they may produce a mutant receptor that doesn't even need instruction from a growth factor to trigger the events inside the cell leading to division, okay? And finally, we may end up with a situation whereby neither a growth factor nor a receptor is needed. The cascade leading to initiation of division can be triggered by other internal factors inside the cell. The The last thing I want to tell you about cancer is that particularly malignant tumors are very heavily dependent on nutrients and energy, which requires oxygen to be produced. And therefore, adequate blood supply is very, very important for tumors to grow and then metastase and further develop. To that effect, they form their own blood vessels by releasing growth factors such as vascular endothelial growth factor that triggers branching of existing blood vessels, which then end up supplying particularly specifically the tumor with nutrients and oxygen for the tumors to be able to grow. Now, let's just talk very briefly about what you will study if you choose to do a course in biomedical science with us. You will, of course, study very many topics in biology with particularly specialist areas in human disease and clinical practice, applied clinical sciences in genetics, biochemistry, immunology, microbiology, and others that I haven't listed here. And you will develop in-depth knowledge of the biology of cancer in principle. I actually illustrated to you what you will be studying even from your first year here at UEL. And you will also further the following years develop specialist knowledge about some specific widely sort of diagnosed cancers that we are going to discuss with you in different modules. And upon graduation, you'll be able to perform and interpret diagnostic tests to diagnose various cancers. If you'd like to learn more about doing a course in biomedical science, this is a QR code that you can scan and learn more about this particular course. If you choose to study biochemistry with biotechnology, you study subjects such as molecular biology, which is very important for cancer because that's where we study about the genes, biochemistry, genetic manipulation, cloning, genetic engineering, and molecular therapies. You develop in-depth knowledge of the molecular mechanisms of cancer development. Yeah, this is about the genes, how they're regulated, how do you make sort of a tumor suppressor inactivated, or how can you abolish that inactivation so they can still do a job, and so on and so forth. And you will sort of be acquiring knowledge about genetic manipulation techniques, whereby the future of therapies in many diseases lies in being able to manipulate specific genes responsible for specific diseases. Upon graduation, you'll be able to develop new diagnostic tests and potentially new gene therapies for cancer, if that's what is your interest. And here's the QR code that you can use to learn more about this course. And if you choose to study medical physiology, you study human physiology, first anatomy, of course, and then biochemistry, pharmacology, and you'll develop in-depth knowledge of the structure and function of cells, tissues, organ systems, and techniques to assess that function uninvasively, not particularly sort of doing anything to a person. And upon graduation, amongst others, you'll be able to perform and develop non-invasive diagnostic tests for cancer diagnosis. And this is the QR code where which you can scan and learn more about this particular course. Now I hand over to Dr. Rahman to talk to you briefly about the chemical and drug sciences.
Thank you very much, Sophia. As my colleague already introduced me, I'll just say very briefly about myself, and then I'll go through about anti-cancer drug, and then I'll discuss about the courses which cover in my subject area. So I am Dr. Mukhlisur Rahman. I'm an associate professor, and I am subject area lead for drug and chemical sciences. Within my subject area, so there are three undergraduate courses. BSc Pharmaceutical Science, BSc Pharmacology, and BSc Chemistry. Right. So before I go about the courses in my subject area, I would like to give you an overview about one very popular anti-cancer drug. My colleague also already mentioned about the different characteristics of the cancer, different types of cancer, their diagnostic feature. Now I'm going to talk about the drug discovery aspect of cancer. So paclitaxel or Taxol is a anti-cancer drug. So this is currently the best-selling anti-tumor agent. So originally this came from a plant. So specific yew, Taxus brevifolia is the source of this compound. You can see this is a very complex compound if you look at the structure. And this is originally the plant was collecting from the Northwest Canada and the USA, where the plant is widely grown over that area. Two scientists in 1960s, they isolated this compound, but it took like a couple of, I think at least 2 or 3 decades to confirm the structure and then to evaluate their anti-cancer properties. So if you looked at this structure, this chemical structure is really, really complex. And you can see, can you just click on this slide, please, just to show how it looks like? Yeah, just click. Yeah, thank you. You can see oxetine ring, which is on the right-hand side, and this particular ring is responsible for the anti-cancer properties. Can you click again, please? Right. So this Taxol or paclitaxel is clinically used for the treatment of the different types of cancer, particularly for the treatment of ovarian and breast cancer, and also non-small cell lung and small cell lung cancer, and also for the cancer for head and neck. So this is very briefly about the anti-cancer drug. When you study here, you learn about more about the different types of drug. It could be anti-cancer, it could be the other types of drug. Can you go to the next slide, please? Right, now I'll talk about very briefly about the 3 courses which cover in my subject area. The first one, pharmacology. So we are offering pharmacology for more than 50 years within our school. In pharmacology, so you're going to study the different topics in medicines, which is related to the use, effect, modes of action of the drug, including their biochemistry, physiology, toxicology, and immunology. Whenever I talk about pharmacology, I give a very simple example. Whenever you got pain, you take the painkiller like paracetamol, aspirin. So pharmacology is about how this painkiller act within the body. What is this mechanism action and why you are given like particular dose? So what are the adverse effect of this drug? So this is all you learn in pharmacology within in 3 years. And also you need to know about the toxicology of the drug. Now, how does it link with the drug discovery? For example, when you talk about the anti-cancer drug, so you learn about how it works. You remember I mentioned about there is a particular functional group which is responsible for the anti-cancer properties. So now what is the overall mechanism of action of Taxol within the body when a cancer patient take this drug, and what will be its adverse effects? So you learn all of those. Now, if you wanted to learn more about pharmacology, and also if you wanted to apply to pharmacology, please scan this QR code so you'll get the detailed information about pharmacology. Can you move to the next slide? Yeah, thank you. So the next course I'm going to talk about is BSc Pharmaceutical Science, which we started in 2012. So pharmaceutical science is also about drug or the medicine. So in pharmaceutical science, we talk about the discovery of drug, what is the source of the drug, where the drug come from, and how do you make the drug? What are how do you or develop a drug? How do you manufacture drug? And also, how do you maintain the quality of the drug? When we are giving the example of paracetamol in case of pharmacology, so in pharmaceutical science, you know what are the source, how do you synthesize this, how do you manufacture this, how do you maintain the quality of the drugs? This is all the things you'll be learning in pharmaceutical science. In terms of anti-cancer drugs, the source of medicine, which we mentioned in my initial slide about it came from the plant, and now how you can make in the large scale. So how can you synthesize these particular drugs? So these are the things you learn. If you wanted to learn more about the course, so please go through this QR code so you can just explore the different area of pharmaceutical science, and you can learn more about this course, BSc Pharmaceutical Science. Next slide, please. But the final course, I'm going to talk about BSc Chemistry. So in chemistry, we cover different areas of the chemistry. Inorganic chemistry, organic chemistry, physical chemistry, and analytical chemistry. So these are the 4 different areas of the chemistry. So we cover all of those. You also learn about the medicinal chemistry, drug discovery, when I talked about Taxol. So what are the methods to synthesize this one in the laboratory? So this kind of thing you learn in chemistry. You also learn about the environmental chemistry, green chemistry. So when we talk about the anti-cancer molecule, is how you synthesize this molecule. So this is the key thing you learn in chemistry. Chemistry is accredited by Royal Society of Chemistry, and pharmaceutical science is accredited by Academy of Pharmaceutical Science of Great Britain. So these 2 courses are accredited by 2 professional bodies, and also BSc Biomedical Science is accredited by the IBMS. All right, now if you want to learn more about chemistry, please feel free to scan this QR code, so which will show you the more information about this course. We are going to finish off here, and I don't think there is any question in the chat. But normally the type of the question when we are doing any presentation, so that the type of question normally student ask, what is the balance between lecture and practical in our courses? So Sophia, do you want to answer that question, or do you want me to answer that? I mean, in terms of, I think you are muted.
There's a healthy balance between lectures and practicals, but you'd have to appreciate that all of the courses that we mentioned here are seriously rooted in laboratory work and clinics. So therefore, people interested in this course, you should be prepared to spend a considerable amount of time in the lab, and should be aware that we take development of laboratory skills, practical skills very seriously, which is the strong sides, the strong features of all of our courses, because this is what employers look for, and this is what employers are interested in. So there's a considerable, serious sort of laboratory staple in all of our courses.
Thank you very much. As my colleague mentioned, it's more or less for each and every module, you need to think about, you'll be doing more or less around 50% of practical when you are studying any course in bioscience. I think there is another question normally the student asks, who is like, how many days I need to be on campus when I'm studying this course? So do you want to answer, Sophia, this question?
Every undergraduate course has 3 modules per semester. These are all 20 credit modules, and they equate to roughly 40 face-to-face hours. Our semesters are 12 weeks long, with a consolidation week in the middle when there are no classes. So that actually works roughly to 4 hours per module per week. So we tend to have our modules on different days, but we try to be kind of helpful and understand the students these days have other responsibilities. They need to also work. They need to take care of families. So we aim to have a single module all the classes taking place over a period of 4 hours. That's either 9 to 1 or 1 till 5, depending on the timetable. And that means 3 days a week, half day, is the bare minimum that students should expect to be spending on campus doing any one of our courses.
Thank you very much, Sophia. So the answer is very simple. We are expecting you approximately 3 days a week on campus. Right, I think we are coming to the end of this webinar. And if you have any other questions, just feel free to contact us or just scan the QR codes for different courses. So then you can get more information about the courses we offer. Thank you very much.
Thank you very much. Thank you. Bye.